Hey, I'm Bryson Carter. We're going to be talking about smell, taste, hearing, how the eye focuses light onto the retina, photoreceptors, what light is being focused onto, and receptor fields, how we can tell lines and crisp edges with our vision. So we're going to start with smell. The I think of in our noses, we've got an olfactory up there that produces smell because it's called olfactory. That smell is produces smell is through olfactory senses. So we've got our nose. It's a pretty sharp nose, and here's an eye up there, right? Just mapping it out. Right up here at the top, right about here, there's an area that in humans is about 10 square centimeters covered in cells, olfactory cells. So cells that can smell things. And how that works, how we can smell things is because of a cascade of events from dissolving chemicals into the fluid up there, and then that causes action potentials eventually. So we'll get there. So these are the nerve. These are like nerve cells right here, and they come up and then they synapse with another nerve cell, and it goes through bone. Here's the bone. This is like epithelial tissue, and so these nerves come up, and then they synapse into this thing called the olfactory bulb, and that is where that's where they all synapse, and then after here they go to the brain. They either go to the olfactory cortex or to the frontal cortex that are associated that associates smells with memory. Smell does not pass through the thalamus, so it's not regulated. It's considered a very primitive sense. So it goes to make strong memories or just strong signals that it's smelling. It doesn't pass through the thalamus. Okay, so we're not going to focus too much on this part. We're just going to focus right here. These nerve cells are actually a very unique set of nerve cells in the body that reproduce or that reform. Normally nerves don't, and if they die, then that's kind of a bad thing because we don't just whip up a new nerve cell willy-nilly, but these ones do. And so that's why certain diseases, they can interfere with our smell, take it away for a bit. But the bad thing is if someone gets smashed in the face and this bone breaks and it severs the axons going from these the olfactory nerves to the olfactory bulb, and it severs them, good luck growing stuff back. You'll lose smell forever, for your entire life. And so don't break your nose up here, but you can sniff paint or whatever, and you will eventually grow your olfactory nerves back. Don't sniff paint, but so we're going to zoom in on so just a rudimentary drawing right here with we're just gonna do some animations. So that should be an arrow. Okay, so we've got like a fluid right here, okay, and chemicals in the air, little tiny chemicals are floating around, and they eventually make it up your sniffer into this fluid, and then they dissolve. And that's really important because without that, they won't be able to bind to the nerve. So these different chemicals bind to a G protein coupled receptor. So yep, we've got a GPCR right here, and so it's a ligand gated GPCR. Well, all of them are ligand gated, and so this is going to cause adenylyl cyclase to create cyclic AMP. And so adenylyl cyclase creates cyclic AMP, which acts as a second messenger on the inside of the cell, and this causes eventually calcium ion channels to open up, causing them to rush in, activating synapse and snare proteins to release neurotransmitters, and that is going to cause an action potential to carry on throughout the olfactory nerve pathway into the brain, not through the thalamus, but yeah, to the brain. So that's pretty much important. The main takeaways, there are a lot more details here, but the main takeaways is that let's see. Yep, it's cyclic AMP. I was thinking IP3, but that's with taste. We'll get there. GPCR from the chemicals that we sniff that get dissolved into the fluid around these nerve cells. That causes a secondary messenger, adenylyl cyclase, to create cyclic AMP, and then cyclic AMP is the secondary messenger, and that goes eventually does some other things, important things, and ultimately leading to calcium channels being opened up. Calcium rushes in, changes in voltage, cause the synapse and snare proteins to induce exocytosis of those neurotransmitters. All right, so that's basically smell. Okay, to save some time, I drew out the taste pathway. So we've got our face. We got Randy right here. Randy's got a tongue sticking out, and we're going to zoom in onto Randy's tongue. And this is the surface of Randy's tongue. So you can actually see these structures on your own tongue if you're sucking on a grape lollipop or something, and then you look at your tongue in the mirror, and you look really closely, you can see these little raised bumps. And there's some other methods that you can do, and different tongues have different amounts of these bumps. And it's interesting because most or females tend to have a lot more of these bumps per centimeter of tongue than certain males do, which is pretty interesting. It's not always true, but generally. So the more of these bumps that you have, the more refined taste that you'll have. So that's classifying different tasters, people's ability to taste. So let's zoom in on this bump. This bump is called a lingual papillae. Think about it like lingual, like linguist, like you speak using your tongue. So it's the lingual, it's on the, and then the papillae, it's just like, I guess, I don't know, there's probably some cool Greek root for that. All right, but it's just like it's a bump, okay? We've got a crevice called the circumvallate papillae, or circumvallate, I don't know, however you say it, circumvallate papillae. That's the groove that surrounds the papillae, making them identifiable, I guess, raise up. Okay, but this groove is really important because if you've got a really dry mouth, a very dry tongue, you can't really taste very much because you need fluids to fill up this circumvallate papillae, the groove, and dissolve whatever your chemical it is that you're tasting. And then here we finally get to what the taste buds are. So the taste buds are these little buds that are attached on the inside of this circumvallate papillae. So you've got about 125 stem cell taste buds on in one of these buds of taste cells. Now, not all of them are active taste receptors. They are stem cells. They're highly proliferative. They produce quickly, and that's so that if you burn your tongue with hot chocolate, the next day you can taste. And so that's pretty nice. All right, so only a few of those 125 stem cells in each taste bud actually work as taste receptors, and this is how that works. So we've got our taste cell. Now these do not have, they don't get an action potential. Instead they call it a graded potential. And if you're confused on what that means, you can reference a video on what action potentials are and graded potentials earlier in the series. Okay, so we've got here a different channel. Now different channels are for different substances. And so we've got, and I guess I should add that each of different taste cells, taste receptors, have unique channels to them. So we've got several different taste cells. I'm just not going to redraw each different type. So assume that the channels are the only things that are going to be changing as I describe them. So we can taste sour, and we can taste salty, we can taste sweet, we can taste bitter, and we can taste umami. That's the Japanese word for delicious. We'll describe that in a second. Okay, so let's start with sour. Sour, we taste sour by the presence of protons or acid. Think about vinegar. That's just has a higher acid content than other things. So we taste that as sour because this is a proton channel, and protons enter the cell, and then that eventually causes a graded potential. So the threshold or the resting membrane potential becomes more positive enough to allow voltage-gated calcium channel, ion channels to open up. Then calcium comes in. It's always going to be calcium at this step on every cell. So calcium then rushes in and induces exocytosis with the snaps and snares in the vesicles of neurotransmitter. And then that neurotransmitter comes into the synaptic cleft and binds to the postsynaptic membrane of the neuron, and then sends an action potential to our brain. And there are different nerves, different nerve names. We're not going to cover that in this video. So that's what happens when sour happens, or when we taste sour things. Now let's talk about salty. So instead of a proton channel, it's going to be a cation channel. Now, yes, it's what I mean by cation is I'm talking like any kind of salt, I guess, like calcium, magnesium, potassium. Okay, so if you've got an ionic compound, we're going to taste salt. I guess lithium. I wouldn't be second on lithium batteries, but so yeah, just whatever is an ionic compound that can rush in as some cations. Cations will rush in. That induces a graded potential. Calcium, once again, rushes in with that graded potential. Exocytosis happens, neurotransmitter releases, and then is when the action potential happens. Boom, right there. Once again, you can watch a video previously describing that. I'm going to move faster now. With sweet, we've got the main ligand for that is, I was going to write sugar, but carbohydrates. Or sucrose. Right? So this is not an ion channel. Instead, this is a G protein coupled receptor. And that eventually will lead, so the binding of this ligand to this GPCR will lead to a second messenger of IP3 to induce some changes and resulting in voltage-gated calcium channels being opened up, right? It has a graded potential, exocytosis occurs, action potential from those neurotransmitters. Okay, so it's the same thing with bitter. The ligand for these, it's like quinones or something. I don't know. I'm not even going to spell it. I'm just going to put QU because, yeah. And so there's 80%, I think it's 80% of quinones are toxic to us. And so this is, that's pretty interesting. But same thing, it's a GPCR, so it's gonna let the second messenger of IP3 to cause greater potential, calcium will come in, boom, neurotransmitter, action potential. Okay, umami, the ligand for umami is glutamate. So a lot of Japanese food has monosodium glutamate. And that's because they want to have that, like, the meaty flavor. It's a flavor of meat. I think it also means umami. I think it also means beef or something like that. But this is, I guess, where we get the taste of something's meaty, or like, oh, there's some good meat there. It's from glutamate. Glutamate acts as the ligand, inducing eventually IP3 as a second messenger to come in, do a bunch of cascade stuff, whatever, calcium comes in, and then action potential. Okay, so these are the main takeaway from taste. So thanks, Randy. All right, so we're going to come over here and talk about smell. So we did smell. We did taste. Now let's talk about hearing. This one is pretty intense. This is not going to be an anatomy description of the ear. So if you want a good anatomy description, there's some good YouTube videos on that. But here we've got an ear, and the ear's structure is designed to channel sound waves into the ear canal. And once again, there's some proper names for that, but we're just going to keep it basic, just with the physiology is what this video is dealing with. So this is the eardrum, or the tympanic membrane. So sound waves are compressed molecules of air. So sound needs a medium to travel through, and that medium is air. It can be also fluid and solid. All right, so we're just going to represent sound waves like this. Okay, so we've got different, like, you can say amplitudes of sound waves coming in. And let's say that this is me saying, like, hello, how are you, whatever. So, like, you can imagine what those lines represent. They're going to come and they're going to vibrate the eardrum, kind of like leather on a snare drum, right? So they're going to vibrate this eardrum, the tympanic membrane. And the tympanic membrane is connected to a couple different bones, which are really cool. They're super small. They don't look like this, but actually let me just cartoon it. Hopefully that looks like an anvil. And hopefully that looks like a stirrup. A stirrup like on a horse, those metal foot holders, they're called stirrups. So that's why it looks like that. All right, and then this goes to the oval window. And to the cochlea. Okay. So we've got sound coming in. It vibrates. So sound comes in, it vibrates the tympanic membrane, and that causes the malleus. I think I butchered the spelling on that, but the malleus, which means hammer in Latin, I think, it vibrates the malleus. Then the malleus vibrates the incus, which means anvil. And then the anvil will vibrate the stapes, which means stirrup. Somebody said stapes, I don't know. I think it's stapes. Okay, so the best way to think about this is watch my finger, like don't look at this part. My fingers are like just moving, like this end of this marker is moving like a centimeter in either direction, right? But with moving that just 1 centimeter on this side it's moving like 4 centimeters in either direction. So it amplifies my movement, the length, right? And that's what this malleus is doing. It vibrates a little bit on this side, so then on this side it's moving a lot. That causes a bigger vibration on the incus, and then same thing, they're not arranged like this, they're more advantageous, like angles and stuff. Same thing, the incus then is vibrating this much on this side, but then this side it's like really vibrating, right? So it amplifies the vibration 20 times. That's what you got to take away. So because you've got 3 different bones all arranged to amplify these vibrations. And we need that because this whole thing, the cochlea, is filled with fluid. And we've got, we're going to cut a cross section, and we're going to look down the cross section of this cochlea. Okay, so I'm now going to erase, actually. Bam. Not bad. All right, so now I'm going to draw the cross section of the cochlea. We've got we've got the scala. I know this is the scala media. Once again, it's not an anatomy course. This has got to be the scala. It'll be nice to refer to these names though, because they have different concentrations of different ions, which is important. Okay, so we're just going to leave it at that. Actually, let's see. Okay. So this is the cross section of the cochlea, and in here, this is going to be the organ of Corti. And in the organ of Corti, we have a gelatinous structure called the tectonic membrane. No, it's tectorial. Tectorial. Called the tectorial membrane. And then we've got down here, this is the basilar membrane. Then right here, we have 3 outer hair cells and 1 inner hair cell. Now I should draw this like this. Okay, so this is really important because the basilar membrane is, if you were to trace this out, the whole basilar membrane goes, and if you were to pull out the basilar membrane, opposite way. According to this drawing, I need to orient it like this. So starting right here at the very front of, closest to the outside of the ear, we've got these fibers that are very stiff and hard to vibrate, and these ones down here are very loose, easy to vibrate. So this corresponds, no, I did that backwards. This should be 20,000 hertz to 20 hertz. The lower the frequency, the lower the tone, the pitch, because the larger the vibrations, or the larger the fibers that vibrate are. So when sound comes in and this vibration occurs into the fluid, because this is all filled with fluid, that vibration will choose one of these fibers to resonate with. And best, or maybe not the best way, but a way to think about this is if you have like a glass Coke bottle and you want to blow over the lid to make like a howling noise, like whoo, whatever. If you blow too fast, it's not going to make the noise, or it will, just won't make the noise that you're wanting. So then you change the speed of your blowing, the wind that you're creating, to a certain speed, and that will create resonance between that. In like the same manner, this same thing's happening here. So we've got resonance is if you've got a sound wave and something is vibrating at the same frequency, then these waves are going to add to each other, and it's going to be louder, or a bigger wave. So that's what resonance is. And so when the sound at a certain frequency hits a certain fiber that is also vibrating at that certain frequency, that is when the magic happens and we are able to hear. Most human voices are around 3,000 to 5,000, I think, hertz. Humans can detect 20,000 hertz to 20 hertz. That's our range. Most of the time, when we get older, we start losing that ability to hear. That's why our parents can't hear, say, an 18,000 frequency, but maybe we do, and it sounds like a really high-pitched dog whistle, right? So that's the idea with frequency and the basilar membrane. But let's go into how our brain interprets the vibrations of the basilar membrane. So the basilar membrane is right here, going this way, right? We've just cross-sectioned it. And all across the basilar membrane, there are these hair cells. They're not actually hair, but they just look like hair. And so that's why they're called hair cells. On the hair cell, we have mechanically gated potassium channels. So mechanically gated potassium channels. And there's a little protein thing that is attached to this side, or the opposite, like between the fingers of the hair cells. And there's proper names for that. We're not going to go over that today. So when this particular area of the basilar membrane finds its resonance with whatever sound wave that's coming in, and it starts vibrating up and down, the tectorial membrane, this gelatinous structure, causes tension to occur in it just pulls on these, I think they're cilia or whatever, these fingers of the hair cells, and that pulling opens up these potassium gated ion channels. Now, who cares about the opening of these potassium gated ion channels? Let's talk about what's happening with how our tissue potential is created. It's not a membrane potential, but it's a tissue potential. Right here, right next to this side of the cochlea, we've got a bunch of cells. Now these cells are covered in a ton of blood vessels, and I'll explain why. But we're going to zoom in on one of these cells. Okay, so on one of these cells, we have a sodium-potassium pump. Good old sodium-potassium ATP pump. ATP. Okay, sodium-potassium pump. And so that's going to be pumping in 3 potassium ions and out 2 sodium ions. And then down here, though, we also have something called the NKCC channel. And this stands for sodium potassium chloride channel. Now, note that I put a 2 in front of the chloride. That's because for every 1 sodium and 1 potassium, it also brings in 2 chlorides because that creates a neutral charge. And that's going to bring all those into the cell. Then on this side only of these cells, so on the outer side, we have a chloride leak channel. And then on this side, we have a potassium leak channel. So check this out. A ton of these cells lined up, all doing this, only having leak channels on these opposite sides according to the potassium chloride. That is going to create a charge separation. And that is why we have a net positive charge right there and a net negative charge right there. On the outer, like this is separated by tissues, not just by a cell membrane, but by cells and tissue. So now we're going to zoom in to this hair cell. So this is the basilar membrane right there. This is the one that's bumping around, resonating with the frequencies coming in, the sound waves coming in, moving the hair cell, rubbing it against the tectorial membrane, causing these mechanically gated potassium channels to open up. Well, on the inside of the scala media, this fluid has a concentration of 150 millimolar potassium, but so does this. So potassium doesn't want to go into this hair cell, even when it's opened up only on, or just solely based on concentration gradients, because it's the same. So potassium's chilling, it's happy, same concentration gradient, but we do see that it flushes in here. Potassium flushes in when this gated channel, mechanically gated potassium channel is open. Well, why is it doing that? It's because on this side, on the outside, remember how we have this tissue potential created by all these sodium potassium pumps over here and the NKCC channels and these chloride leak channels and potassium leak channels positioned strategically where they are. Well, that's creating this massive charge difference. And in here, this is this has a driving force of negative 40 millivolts, or I should say it has a membrane potential of negative 40 millivolts. And on the outside, we've got a potential of negative 80 millivolts. Add them together, you get a total of negative 120 millivolts of a potential separation. And voltage is really just like I said, the potential that's going to happen, or what could happen, the movement that is available. And so once these open up, potassium does want to come out here because of the electrical gradient, not the chemical gradient in this case, but it's the electrical gradient. So Potassium is going to rush in and it's going to rush out. Now that is going to allow, and I should put these negative charges in here too. That is going to allow this hair cell to depolarize, which causes snaps and snares to release these vesicles of a neurotransmitter, and that neurotransmitter comes down to this, into the synaptic cleft, to this auditory nerve, and or this neuron, which goes to the auditory nerve, and bam, an action potential occurs, and that's how we hear. This action potential goes to the brain, and the location where it's coming from, the different frequencies of this action potential, all that go into our brain's ability to interpret what that sound sounds like. So there's over here, there's a few more channels which are important, and I should write those down. Let's see here. Okay. So the reason why there's a negative charge in here is because of our handy dandy nifty sodium potassium pump. So that's why it's negative 40 millivolts on the inside of the hair cell. So that's really important to note. And then a potassium leak channel is also right here. Okay, so those are some pretty important key players that will answer, well, how do we get negative charges on the inside of the hair cell? Well, sodium potassium pump. Okay, that's creating the negative 40. This one, let me make sure, yep, it's a potassium leak channel right there. And so it's pretty incredible because this tissue potential has been created allowing us to hear. Now, why is this called the stria vascularis? Well, that means striped vascularis. Like there's a lot of vasculature and it's striped because of it. Under a microscope, that looks really striped with all the blood vessels. It's so densely supplied with blood vessels that people actually think that it's the most dense area in the body that has that many blood vessels going to it. And that's because of all these sodium potassium ATP pump. They need a ton of ATP, so they need a ton of mitochondria, and those mitochondria need a ton of oxygen, so we need a ton of blood, right? It's all going together, why there's tons of these. Some animals have many more layers of these cells. Humans, I think, have 2 or 3. Okay, so that is... Okay, something that's really important to note is that only 5 to 10% of these outer hair cells are innervated, and the rest of the innervation goes to these inner hair cells, which actually aren't touching the temporal or the tectorial membrane. But what happens is when this action potential occurs, it's going to activate, and this one I'm not entirely sure on the details, so I'm just going to kind of crudely draw it up here. It's going to activate this thing, this like, I think it's like a protein or this muscle thing, and that's going to cause it to contract. I don't think it's a muscle, but it does contract. It's a contractile thing, and that contraction of it, by the activation of these hair cells will pull the basilar membrane and the tectorial membrane closer together so that this inner hair cell can come and do the same thing that these outer hair cells just did. Can come up, rub against the tectorial plate, cause these mechanically gated potassium channels to open up, boom, and then and then it depolarizes, sends an action potential down this nerve. That's really important because these outer hair cells amplify. Once again, it amplifies whatever vibration is that's going down. Pretty crazy. So why couldn't the brain just come up with more nerves to these outer hair cells? Why does it need to amplify it? I don't know. But I think it is pretty neat that it allows, perhaps it allows the signal to come in quietly, or maybe it's more quietly, maybe it's more strongly. I don't know. All right, so it's really important, really cool. Okay, so what we're going to talk about now that we've done hearing, we're going to talk about focusing light. So for that, we're going to draw this eye. Yeah, not bad. And we're going to draw so we've got our cornea right here. We've got our sclera, the rest of the eyeball. And then there's a ton more structures, but we're just going to focus on the ciliary body, or the ciliary muscles. And then these are the suspensory ligaments. This is the lens. All right. Now it's important to note right here. Actually, I don't know if it's really that important to note in this situation, but this is a cross section. This represents our iris, the colored part of our eye that gives us blue, green, or brown eyes. And that can, so that's important to, actually, I don't know very much more about that, but, so I'm pretty sure that's the thing that contracts and dilates to allow more light or less light in. But let's talk about how light is focused. So we have, here's the retina, the back of the eye where light is focused onto, has a ton of these little rods and cones, and the center is called like the linea macula or something like that, and that's where the fovea is, where it's the highest concentration of these rods. Pretty sure it's rods. So what happens is light enters and then it bends and then it comes ideally and focuses right, bends right on our fovea, or right on our retina. Sometimes though, if our lens is messed up by our muscles being messed up, then that point of focus happens before it reaches. And so that's where our focus is, because of how the lens is shaped at this particular pathology of the eye. And so that prevents that creates a blurry vision. I just think that's kind of neat to realize that, oh, it's the point of focus is off. Okay, but that's just kind of good review. The main thing that I want to get to right now is what happens with the ciliary muscles. So let's just come over here and draw. This is convex lens, and I want you to think I should draw like this. I hope that you will not get confused anymore between what convex and concave lenses are. So concave lenses are like that, and convex lenses are like that. I really like visualizing the structure of the word. This only works in English, obviously. I don't know what they are in other languages, but this is important because, and this is a little bit of physics, and there's a lot more of this to learn, but when you're driving down the road and you hit the rumble strip, it might be hard to notice this with the rumble strip, but it tends to pull you slightly that way. Or how about when you're driving down the road and one side of the road, it was just raining, so one side of the road has a puddle, and you hit the puddle with those tires, your car wants to turn into that puddle. And that's because the friction that happens when you hit that puddle is more than it is for the road. And so that friction, that uneven friction, tends to turn you to wherever that puddle is. So in kind of like the same way, it's not really friction, it's called index of refraction, or like the indices, the different indices of materials. So let's just talk about glass. Glass can bend light, and that's because it has different, I think I'm just going to throw out some arbitrary numbers. Let's say that air has an index of refraction of 1, but if a lens has a higher index of refraction, that's like us saying that it has higher friction than the road. And so imagine our little car traveling down, I should put it like this, traveling down this road, and it's coming down, and then what's going to hit the puddle or the puddle or the lens first? This side or this side? It's going to be this side, because that's how it's shaped for a convex lens. And so because it hits this side, this car is going to want to turn into it. That's like the car that wants to continue turning into the puddle. So this whole thing is going to be shifted like that. Okay, so that is really to me, that's the best way of conceptualizing how convex lenses bend light. Now, same thing though with concave, is they're coming in this car. Well, what hits it first? It's going to be this side. That side hits the glass, the puddle first. The difference, the different index of refraction first from what the medium that it was traveling in was before. All right. So now that we kind of get that idea, convex lenses are going to bend light like that. And I'm just, drawing it to exaggerate it like that. Concave lenses are going to draw it, or are going to bend light like that. Okay, so with our eyeball, we have a convex lens most of the time if we're looking at things that are close up, because well, if something's far away, then the angle of light between the two doesn't really need to bend all that much. And so our eye wants to straighten out the lens so that it can just go straight into, that increased. But since they're so close together, it's already basically focused. And so our eye doesn't want to bend the light anymore. And that's with objects that are far away. When you are looking at something a lot closer though, like the smiley face, and your eyeballs right here, these light rays are coming in, and here we'll draw this is the lens right here. These light rays are a lot farther apart, and so your eyeball accommodates or bends the lens, or bends the light, so that instead of being so far apart, these light rays are now bent to allow a focus point. Whereas before with the long distance, that point of the light is already basically focused, so your eye doesn't need to work that hard to focus it. And so the flatter the lens is, because it doesn't want to bend the light so much. But the closer the object is, the more oblong, or the more football shaped that lens is, the more convex that lens is going to be. How does the brain how does the eye control the shape of the lens? Well, it uses ciliary muscles, which attach to ciliary or suspensory ligaments. So instead of a cross section of the eye, I'm going to draw a full-on, like a portrait of the eye, or face shot of the eye. So there's the lens. These are suspensory ligaments. And then this is the ciliary muscle. Now here's where the biggest confusion comes in. When the ciliary muscle contracts, which way does it go? So when the ciliary muscle contracts, you might think that it's going to get bigger, but it's actually getting smaller. It's like almost like a sphincter. The ciliary muscle, when it contracts, it's going to get smaller. If it gets smaller, then these suspensory ligaments are not going to be as tight. They're going to be looser. And then this now I'm confused. Because I'm about to teach that since they're looser, that the lens is going to be more football shaped, more convex. But I think that's wrong. Okay. Nope. So I am right. I'm just a little bit confused. Yep. Okay. I don't know. Yeah. All right. So when the ciliary muscles contract, then these suspensory ligaments get looser, which causes the lens to get more convex or more bulged. Now, when the ciliary muscles relax, they go out. That pulls on the suspensory ligaments, pulls that out, which are attached to this lens. And if you pull on these ends, then this lens is going to come like this. So by default, that is, if our eyes are relaxed, we're seeing far vision. Because our lens is, we're seeing far vision better. Because our lens is not convex, it's more closer to concave or more flat. Okay, so that's really good to understand. Pretty neat that the eye can do that. And so that's the focusing of light. And here we go. We're just going to be a long video right here. So light gets focused right there on what happens when this light hits the photoreceptors on the retina. This is pretty darn cool. This stuff is nuts. So I'm going to just draw a rod cell. So this is a rod, and we've got cones in our eyes as well. Cones see color, rods see black and white. Rods are in more abundance. They are very sensitive at night, and they do see color. So this is the rod, and this is the bipolar cell. This is the horizontal cell, and this is the nerve. So we're going to zoom in on one of these photoreceptor cells, or photoreceptor parts of the rod cell, and we see that this is a non-specific cation channel. And okay, so in here there is a in transducin, there's a molecule called 11-cis retinal. So 11-cis retinal has, I'm not quite sure what it looks like, but, and I'm not counting these carbon bonds, but I'm just representing them right here. So these are all trans configurations because they're opposite of each other, but then cis, which means same, that same side, that has a double bond, which has pi electrons, which can be excited by different types of energy, and in this case, it's the electromagnetic spectrum, the energy that we see. So light comes in and excites these pi bonds and creates a 11 trans. You could also say it's all trans retinal. So 11 trans retinal the shape that changes, that the opening of this tail has some effect of it goes and activates phosphodiesterase, PDE. Now, without this activated, this cyclic GMP ion channel is just open always, letting these cations into the cell, depolarizing this cell, this photoreceptor cell, and yeah, it's depolarizing it. And now we're going to break down why or what happens when it depolarizes. But first, let's focus on this. Phosphodiesterase, once it's activated by 11 trans retinal, phosphodiesterase goes and just chops cyclic GMP up. Just chops it up, so no more cyclic GMP, it just turns into GMP. And that way, this nonspecific cation channel turns off. It shuts. Cations no longer enter the cell. Well, before, cations were entering the cell, creating enough of a greater potential to induce calcium to come into the cell to induce snaps and snares, to release glutamate, the neurotransmitter glutamate. That's what was happening. But then light comes along, ruins the party, tells 11 cis to straighten up, to turn into 11 trans retinol. That causes PDE to get all upset, goes and ruins cyclic GMP's party, turns off this nonspecific cation channel. Those cations no longer excite this whole cell. Light, it's like flipping the lights on at a disco party. Everybody's just ticked, right? Because calcium no longer is coming in. Glutamate isn't being released into the synaptic cleft between the photoreceptor and the bipolar cell. So, why? Who cares? What happens next? Now I'm going to draw a more simplified animation to really get this stuff down. This stuff gets pretty nuts, pretty insane. So we've got, oh yeah, I like that marker. Okay, so this is our bipolar cell. This is our photoreceptor cell. This is also a photoreceptor cell and a photoreceptor cell. This is our horizontal cell. Cells. This is also our horizontal cell. All right, this, now I'm going to recap. So 11 cis retinal goes to 11 trans retinal. That causes phosphodiesterase, and that causes, let's put light. Phosphodiesterase turns cyclic GMP, and it just chops it. And so this nonspecific cation channel is out of the game, which is not releasing glutamate. So glutamate goes down. You will see that there's a general rule that light decreases glutamate. Period, the end. All the time. Everywhere. Okay? Well, let's not say everywhere. Just all the time, light decreases glutamate. Okay, so, because I'm sure, so in the eye, okay? So light decreases, so glutamate's decreased because light just turned on the lights at the party, and that no one's excited anymore. So these vesicles aren't releasing glutamate anymore. Well, what happens is the difference occurs in the bipolar cells. So if you can imagine, copy and paste this right here. I'm just going to draw a different bipolar cell. We have on here a channel called AMPA. AMPA likes glutamate. It loves it. If glutamate binds to AMPA, then AMPA lets in sodium. Sodium comes and opens up calcium, voltage gated calcium ions, and that lets glutamate move onto a nerve, sending an action potential. All right? So, but what activates AMPA? It's glutamate. So AMPA is it's a ligand-gated ion channel that allows sodium to enter in the cell to depolarize this bipolar cell, causing more calcium to enter, causing the vesicles to release exocytosis, right? Glutamate, increased action potential sent. So, but what happens? It's all about glutamate right here. If light comes in, ruins the party in the photoreceptor, flips on the lights at the disco party, glutamate is going to decrease. No longer will this cell, photoreceptor cell, release glutamate. So what happens? Glutamate no longer is going to activate the AMPA channel, and that's no longer going to depolarize the cell. This is no longer going to release glutamate. The bipolar cell will no longer release glutamate, and action potentials will not get sent to the brain. So, because it's not getting sent to the brain, we're going to call this off. And since it's the center cell that had this happen to it, we're going to call it center. Let's just label it center. What caused this off, this turning off of this bipolar cell, which of this eventual neuron? It was light. Light shut off those action potentials. Really interesting. So an off center won't send action potentials when there's light on it. But if the light goes away, then the whole party resumes. The disco gets back going, right? The glutamate, the cyclic GMP is able to bind to this this nonspecific cation channel that allows the sodium, like the cations, to come in, depolarize it, allowing those glutamate vesicles to come. The vesicles of glutamate neurotransmitter to release. Glutamate activates AMPA. AMPA starts being like, hey, I'm happy now. I get to have these cation channels. The sodium come in, calcium comes in, glutamate releases, action potentials are sent. That's what happens in the dark. That's why we call this off-center with AMPA. AMPA is the determining factor, whether it's an off-center cell. Now, let's talk about an on-center cell. This stuff is dense. Nope, it's calcium. Yep, okay. So, mGluR6, glutamate binds to mGluR6. If mGluR6 is activated, by the presence of glutamate, then it sends a secondary messenger that goes and closes TRPM1. It's kind of like Trump1, but it's just TRPM1. That TRPM1 is a calcium channel. And calcium wants to come in. In fact, this is always going to be open unless mGlu6 is ruining TRPM1's day. So mGlu6, if it's active by the presence of glutamate, it's going to cause a secondary messenger to close TRPM1. And so if it closes TRPM1, then calcium is no longer getting into the cell. It's hyperpolarizing this cell. And so these vesicles down here of glutamate are not being transferred into the synaptic cleft. And so then this nerve is not getting action potentials if glutamate is active, if mGlu6 is active. Now let's back up. What causes glutamate? So glutamate is in high abundance when there's no light. So when it's dark, 11-cis-retinal is just chilling in transducin, that protein on the photoreceptor. Okay, it's just chilling there. And 11-cis-retinal is not changing, okay, if it's dark. So that means phosphodiesterase is not active. That means cyclic GMP is able to be cyclic GMP. And cyclic GMP is a binding to this non-specific cation channel, and that's allowing cations to come into the cell, sodium, calcium, and that causes the depolarization of this photoreceptor cell, and that allows glutamate to be released into this this synaptic cleft between photoreceptor and the bipolar cell. And I guess I don't know if it's a synaptic cleft because these aren't really nerve cells, but whatever. Okay, so it's released into the space. So glutamate then comes, and we're over here, glutamate binds to mGlu6, and then that's activating the secondary messenger, which closes TRPM1, that calcium channel, and it shuts that down. And so these vesicles of glutamate down here are not being not being released because calcium's not coming into the cell to depolarize it. This is a non-excited state of the cell when darkness is occurring. But let's check it out with light. Light comes in. If light comes in, it's going to cause this 11-cis-retinal to change conformations into 11-trans-retinal. That conformational shift activates, well, it really activates the GPCR, which sends the alpha subunit, which does some magic and creates phosphodiesterase in its active state, or it signals phosphodiesterase. Okay, so there's some alpha subunits involved, but GPCRs, whatever, right? So ultimately, phosphodiesterase gets activated. That goes and just chops cyclic GMP. It just cuts it into GMP. So no longer can cyclic GMP allow this non-specific cation channel to be open. So with that closed, this cell is no longer being depolarized, which would have released glutamate. So it's no longer releasing glutamate. So glutamate is going to go down. Well, less glutamate means that mGlu6 is not activated. Since it's not activated, this secondary messenger is not going and ruining TRPM1's day. In fact, TRPM1 is like, yes, I'm open now. I'm sending calcium in, full send calcium, deploy, deploy, deploy. And that's all because glutamate is less because of the light. So calcium comes in, and then it depolarizes this bipolar cell, and that allows these vesicles of glutamate in the bipolar cell to release into the synaptic cleft between the bipolar cell and this neuron, and then that neuron goes, should be neuron, not nerve. Neuron goes to a nerve, and the visual nerve, whatever it's called. Anyways, that sends an action potential, and that's why if light hits a photoreceptor that has a bipolar cell that has mGluR6, it's going to send an action potential. So this neuron would be it would be on when light is hitting it. And since it's in the center, we're going to call it center, on center. Boy, that's dense. So, these are named by what light does to the neuron at the end of the day. Okay, so light causes an off-center photoreceptor to not send action potentials. It will be no longer sending action potentials. But if light is doing that, if light is preventing action potentials, I gotta dance so that this light turns on. I don't know why it's getting so dark. Wake up. All right, we're back with the lights. So light, if light shines on an off-center photoreceptor, then it is no longer sending action potentials. But if there's no light, then it's going to be sending action potentials. So in an on-center, if light hits the on-center photoreceptor, then action potentials will be sent, like one might naturally expect. So that's that. But we haven't even spoken about receptor fields and horizontal cells. So let's jump into that. Whew. This is quite dense. It's pretty cool how they found this out. I think that's pretty neat. The research that they did, maybe they're all just making it up, but no, I don't think so. I think this is really neat. And it's really cool how the creator has set some things up and allowed evolution to create these things, and yeah, that's really cool. Okay, so this photoreceptor is still like this photoreceptor, but it's on the sides, and if light hits this, light is going to, well, let's first talk about absence of light. Okay, we have glutamate coming out. Same thing, right? 11 cis-retinal to 11 trans to PDE, phosphodiesterase, cyclic GMP, right? So if none of that is working, cyclic GMP is happily allowing cations to come in, depolarizing this cell, which allows glutamate to be released by exocytosis. Glutamate is going to come to this horizontal cell, and guess what is on the horizontal cells? All horizontal cells have AMPA channels. Well, we already know what AMPA likes to do. AMPA likes to bind to glutamate and cause depolarization of the cell, cause those sodium ions to come in. So sodium is going to come in. The difference is what releases? It's GABA. So GABA is a new neurotransmitter, and GABA is going to come over here Historically, GABA is seeming like an inhibitor in the brain, so it likes to inhibit things. Um, so, but GABA, and you'll see that it does that here, GABA is going to come bind and open either chloride leak channels to allow chloride to rush into the cell, or it's going to open up potassium leak channels to allow potassium to leave the cell. Either way, the net result of GABA on the center photoreceptor is hyperpolarization. So, or in other words, it's moving towards that more. So it's going to, it's kind of like turning on the lights at the party. It's hyperpolarizing, excuse me, or reducing the depolarizing nature of this cell presently. So that's what caused this GABA to release? Well, it caused darkness. It caused darkness to be, darkness allowed glutamates to be in high presence or high concentration, which allowed glutamates to bind to AMPA channels on the horizontal cells, which allowed it to depolarize enough to send GABA, that neurotransmitter from these horizontal cells, into this area, which bound to the photoreceptor center cell, which allows chloride to rush into the cell, or potassium to rush out of the cell. Either or, the net effect is the same, and that is reducing action potentials from that one. Same thing happens over here if this is darkness. So, we are going to throw up some arbitrary numbers. This is not actually true, but we're just going to throw up some arbitrary numbers to represent kind of the net effect of how this whole thing is a receptor field. This whole setup right here. This is a receptor field, and we're going to describe how the brain senses corners, sharp lines, edges, stronger. So we're going to do that by representing a few numbers here. And a much simpler drawing than this even simplified drawing. So this center circle is represented by this center photoreceptor. So we're going to do, since this was an off-center, we're going to color it dark. Just by convention, we're going to do that, and you'll see kind of how we can match it up. So, and then here's just a general rule of thumb. If something is off-center, that means the surround photoreceptors are going to be opposite of whatever the center is. So if something's off-center, it's going to be on-surround. And so these would be on-surround photoreceptors. Now what does on mean again? On means that if light hits the surround, on-surround, if light hits the surround of an on-surround photoreceptor, then an action potential will be sent. But if light hits the center of an off-center photoreceptor, no action potentials will be sent. And I literally just described why with this off-center. Okay, so we're going to just write up some numbers here. 2, 2, and then this middle one is 6. If you add them up, it equals 10. And that's a simplified version of 100%. So we're going to say that these surround photoreceptors have about a 40% influence on whether the action potential, or a 40% influence on the signals of the action potentials sent to the brain, while the center has about a 60% influence on the action potential sent to the brain. So, in this case, we are going to shine, I'll just use this red, okay. We're going to shine some light only on the center photoreceptor. And in the absence of light, you have darkness. So I'm just going to still draw that like this. Okay, now these are on surround because it's with an off center. And if light is hitting the off center, we already know that no action potentials will be sent. So out of the 6 possible action potentials, or like the 60% influence, so let's just say there's 6 action potentials. Out of the 6 possible action potentials, none of them are being sent because light is hitting the center of an off center. Remember, like this whole thing, light ruins the party. It decreases glutamate. Glutamate was depolarizing this cell, allowing it an action potential to be sent, but it's no longer sending that. So if light's only on the center, nothing's happening. Well, and here's why we designate these surrounds as on in the case of this. Because let's replace this with light. Okay. So if light is hitting both the center, so before with darkness hitting this on surround, it's not going to send any action potentials, but that's going to be a little harder to explain, or it's harder to understand. So we're going to now explain what this is with the light. When light hits this surround, so we already know that if light hits an off center, we're not going to get these action potentials. And if light hits this surround, though, let's walk through. Light hits the 11 cis. This might be confusing you, so I'm just going to draw it up here. 11 cis, causing it to turn into the 11 trans, which causes phosphodiesterase to go and chop cyclic GMP, which closes this nonspecific cation channel, which hyperpolarizes the cell. So now glutamate is not coming into this area. That is not happening because it's hyperpolarized, so glutamate is not getting released by exocytosis. So remember, AMPA really likes glutamate on the horizontal cell, but with decreased glutamate, AMPA is no longer binding to glutamate, and that's no longer depolarizing the horizontal cell, which is no longer releasing GABA, the neurotransmitter GABA, into this area. And GABA is no longer coming here, causing chlorides to enter into this cell. And when chloride enters this cell, it depolarizes, it hyperpolarizes this cell. But if that's not happening, if chloride is not going into the cell, then this cell will still release glutamate. And glutamate, remember, when there's glutamate in this area, glutamate binds to AMPA. And AMPA is like, hey, at least we can have a party down here. AMPA full sends sodium into this bipolar cell, which causes calcium to full send it into this bipolar cell. That allows these snaps and snares, exocytosis to happen, glutamate is increased, this nerve or this neuron is sends an action potential. Crazy, crazy. So what we would do is we would get these action potentials, and we're just going to arbitrarily say that it's about 20%. Excuse me. So 2 action potentials. Would we get any over here? Well, no, because light, like darkness, allows glutamates to be in high abundance, which allows AMPA and to depolarize this horizontal cell, and that allows GABA to be released, which allows chloride entering this cell or potassium to exit the cell. Either way, same effect of hyperpolarizing this cell, so preventing glutamate from coming into this cell, this bipolar region, and with less glutamate, that means less depolarization of the bipolar cell, and that causes less glutamate, and that causes less signals being sent through the less action potentials through that neuron. So this side is not going to, we will not get those 2 action potentials because it's in the dark. Whew. So this, out of 100%, we're only getting 20% of a signal sent to us, because we're only getting light on this, well, because of the whole setup. Now, the best way, like, why is this important? Who cares? It's challenging because, well, no, it's not that challenging. We can explain it. This is pretty nuts, and it took a lot to learn it so far, and I'm not that good yet at it. So all right, so I've drawn these circles. So that's red is light being shown on this. The dark center means that it's an off center on surround. So that means if light shines on the center of it, it won't send those action potentials. But if light shines on the outside of it, it will. And I just explained why over here. So all right. So this is a this is a receptor field, right? The center photoreceptor and side photoreceptors, horizontal cells, bipolar cell, neuron to the nerve. So the red represents the light shown on this whole receptor field. And keep in mind that this is at the back of this is the retina, the back of our eye. And so, all right, let's jump into it. So this is dark center, so we're going to call it an off center. In fact, these are all off center, which mean they are on surround. And with this, I'm going to represent the number of action potentials by lines. So the center is an off center. So that means when light hits, this is the light hitting the whole receptor field, we have light with, remember, an off center has AMPA in the bipolar cell. So that means that it was a ruined party. Okay, so this is not going to send action potentials. The center, photoreceptor, will not contribute action potentials. So, but what about the sides? Well, we decided, remember this story right here, these two? Because this was, this reduced glutamate, which reduced GABA, which reduced depolarization, or hyperpolarization, which allowed depolarization to occur. So that contributed 20% or 2%. So we're going to do 2 from that side. Well, the same story is happening over here. So there's going to be 2% there. So add them together. We have 40%, or we're going to have 1, 2, 3, 4 action potentials in this situation with light hitting the whole thing. Okay, how about here? Well, we remember this was literally this example. Darkness on one side, light on the center, and light on the far left. Well, darkness on one side, light on the center, light on the far left. Remember, we had 2, so 20%. So from this situation, we only had 2%, or 20% of excitation going to the brain from this receptor field. All right, now this is where it kind of gets really it gets cool. So, well, I'll save that for the last. Okay, right here, light's not hitting the receptor field at all in an off-center on surround. So in other words, light's not turning on the surround, so we're not getting those two or those two because it's just darkness on those surround. But check it out. AMPA is able to have its disco party because there's no light. No one's flipping on the lights in the gym or the dance floor. So these are going to have those 6 action potentials because that really leads to increased glutamate. AMPA's happy. It's sending glutamate into the neuron. So 1, 2, 3, 4, 5, 6. All right. Here's where the magic happens. This is the entire reason why we need photoreceptor fields. What's happening? Well, no light is hitting the center. Great. They can have the disco party. Glutamate sent. Action potentials are sent because it's an off center. So those are going to give, this is going to give 6. The center photoreceptor will contribute about 60% towards action potentials. And how about this side? Do we get these two? Sorry, let's write it right here. Do we get these two? Well, it's an on surround and it's in darkness, so we're not going to get those two. Nope. And that's, same thing up here. Okay, same thing right here. They're in darkness. They normally are contributing action potentials if there's light, which is the on surround. All right, but we do have light right here at the one corner of the receptor field. So are we going to get those 2 action potentials? Well, we can review it. Yes, we are going to get them, but why? One more quick time. Light's allowing 11 cis to 11 trans. It's allowing PDE to go CHOP cyclic GMP. That closes that nonspecific cation channel. That causes hyperpolarization, which reduces glutamate. So glutamate's not coming out of this far left photoreceptor. Decreased glutamate means AMPA's not happy because it's not binding to glutamate, and that means it's hyperpolarizing, which means it's not releasing GABA, which means chloride is not entering the cell, which means this cell is not being hyperpolarized, it is being depolarized by everything else, And there's more glutamate, which means that there's more glutamates to bind to AMPA, which means that there's more sodium coming in, more calcium, more exocytosis, more glutamate to the neuron. Bam, action potential. So, yes, we are getting these two. So we've got 1, 2, and then 3, 4, 5, 6, 6. Okay, so we have 80% of a signal when light is just on one corner of our receptor field. This allows that, like this means, here's 60%, so we can compare the numbers, 40%, 20%. Interesting, right? This means that our brain receives the most strong signal when it identifies when light is only at the corner of a receptor field. Now think about why this allows us to see lines. Well, it's because light and darkness are right next to each other. Well, what do we get here? Light and darkness are right next to each other. That's pretty cool. That's insane. So that allows us to get more 80% of those signals contributed, and holy camoli. Okay, so I think that's everything. This has been a very long video. This is very fascinating. I did not spend too much time trying to master the nitty-gritty details, the anatomy of the ear, but that this is all about, this has been just about physiology, okay, the actions. And yes, that does include names, but I hope you enjoyed it. This is just me practicing teaching.